Everyone, welcome back to my educational channel. I'm Teacher Janet. In this video, we'll discuss a biology paper 3 question based on the SPM trial exam for 2023 molecular state, right? So this topic is about diffusion across a selectively permeable membrane. The exam question shown here is based on this topic of diffusion, which is found in the chapter 3 chapter of Form 4 Biology kssm syllabus. So in this experiment, which was tested as a practical question or UJIAN AMALI, the student is instructed to prepare a whisking tubing containing a substance, which is actually the substance Y. Sometimes it's stated as solution Y in this case, and it's actually not a solution, it's a starch suspension, right? It could be any type of substance, right? The idea is to find out what kind of substances can diffuse through the whisking tubing, which acts as a selectively permeable membrane. Okay. Now next, the whisking tubing is tied up at the top, and then it is placed in a solution in the beaker. Okay. In this case, it's iodine solution, and this whisking tubing is immersed in the solution for 15 minutes. After that, the color changes are observed. Okay, color changes in the whisking tubing and in the beaker are observed if there are any color changes. So let's find out more about this question and how to answer the questions on hypothesis, inference, table of results, and prediction questions, right? So it will sharpen your techniques of answering these questions and prepare you for the UJIAN AMALI or practical test, okay, which is biology paper 3. Go through the instructions for this front page, on this front page of the biology exam paper. So biology, UJIAN AMALI or practical test, which is 45 minutes, okay, in length. The code is 4551/3. Sebelum diberi masa 5 minit untuk menyimak radas dan bahan yang disediakan sebelum menjalankan ujian amali, kertas peperiksaan ini hendaklah diserahkan pada pengawas amali pada akhir peperiksaan. Okay, so take note that the time to plan and also check your apparatus and materials is 5 minutes before you start carrying out the experiment. Okay, so masa menjawab 40 minit dan markah ialah 15 markah, 15 marks. Okay, let's look at the candidates checklist. You are not allowed to work with the apparatus in the first 5 minutes. This period is used to check the apparatus and materials list, to read the question and plan the experiment, which will be carried out later. Mark, tick, now should be mark, tick in the box provided. Okay, mark, tick here to check that the material and apparatus has been prepared and supplied to you. Okay, so these are the apparatus and materials. A bottle filled with distilled water, 250 ml beaker, Ziploc plastic bag containing 2 threads with 15 cm length each, 5 ml syringe, a glass rod, whisking tubing, which is 15 cm in length, in a 50 ml beaker. Now, concerning the whisking tubing, the use of the whisking tubing, there's a tip here. So first you have to take the whisking tubing out of the beaker, and then you have to wet it in order to open it up, right? So it's like a long tube. Whisking tubing is like a long tube, but it has 2 open ends. Now you want to fold, and then you want to tie one end up to make sure that there's no leakage of contents from the whisking tubing through the tight end. First, fold up 1 cm of the end of the whisking tubing, okay, and then tie this part with a thread below the end here. Okay, tie this end here with the thread. So that means you have folded it up, folded the end up, and then tied it up with a thread, and this will prevent the leakage of contents of the whisking tubing. Okay, rather than just not folding it and tying the end. Okay, sometimes if you don't tie tightly, then the contents can leak from the end. Okay, but if you fold it up first and then tie at the part that is folded, below the part that, I mean above the folded line here, okay, but below the end of the whisking tubing, you tie it up in this section here, then it can prevent the leakage of the contents of the whisking tubing. Next, number 7, solution X in a dark bottle. So this solution X is actually iodine. Okay, if you observe it, it's yellowish brown in color. It's actually iodine, as you will find out later. And then solution Y in 50 ml beaker. So this is a suspension and actually is starch, right? So from the results of the experiment, you will know that solution Y is actually starch, okay? And then you have a wiping cloth to wipe the table, and a stopwatch to time 15 minutes for soaking the whisking tubing. So after 5 minutes, you can start your experiment. Let's read through the instructions here. You have to carry out an experiment to investigate the movement of substances across a selectively permeable membrane for 2 different substances, which are iodine solution and starch suspension, right? So from here, you should be able to conclude that X is the iodine solution because of its yellowish brown color or brownish yellow color. Don't state it as a brown color or yellow color. Sometimes it's not accepted. Okay, yellowish brown or brownish yellow. Now, so we have here we have to prepare 200 ml of distilled water plus 5 ml of solution X, which is the iodine solution, mixed into it. So we can imagine that the color will turn to yellowish brown. It will be a yellowish brown solution. And then in the whisking tubing that we will prepare afterwards, we have 10 ml of solution Y, which is the starch suspension. Okay, it's not mentioned here, but you can conclude from that later on. Then, based on the arrangement from diagram 1.1 here, carry out the experiment based on the following steps. So let's discuss the procedure. Firstly, remove the whisking tubing from the 50 ml beaker and then wet it and open it up, and then tie one end of the whisking tubing with a thread. As I said, fold it up first and then tie it. Then it will prevent leakage. Fold up the end first, 1 cm of the end, fold it up and then tie it up. Now make sure the whisking tubing is tied tightly. So this is a precaution. Tied tightly to prevent leaking. All right, next, number 2, using a syringe, fill the whisking tubing with 10 ml of solution Y. Okay. Now solution Y is actually the starch solution, as you will find out later when the color changes to dark blue, right? So fill up the whisking tubing with 10 ml of solution from the syringe, and then you tie the other end tightly with a thread, right? Next, as another precaution, rinse the outside of the whisking tubing here with distilled water to remove all traces of solution Y, which may have dripped on the outside of the whisking tube, on the external surface. All right. Now next, number 4, pour 20 ml of distilled water in a beaker and using a syringe add 5 ml of solution X. Okay. So now you're preparing the solution in the beaker. You mix 200 ml of distilled water with 5 ml of solution X, which is actually the iodine solution, right? And then stir the mixture with a glass rod to make sure that the iodine is evenly dispersed in the water, right? So the color should change to yellowish brown, as shown here, or brownish yellow. Now, yellowish brown color, the description is based on the SPP answer that was given for one experiment which is similar to this. So I think yellowish brown is okay, but don't describe this color as yellow or brown, right? Usually, sometimes you don't accept that. So yellowish brown. Now, so after that, you record the color of the solution in the beaker, yellowish brown, and then that of the whisking tubing, it is cloudy, whitish, and it's cloudy. Okay, that is the starch solution. Next, place the whisking tubing in a beaker and leave it for 15 minutes. Observe the color produced in the beaker and the whisking tubing after 15 minutes. Okay, so place this whisking tubing, which you have tied up, right, into the beaker. Now try to get the other end exposed at the top so that it doesn't, it's not soaked into the the solution, because if you don't tie tightly, then the solution can leak out from this end. Okay, so we have to wait 15 minutes and then observe the color changes if there are any. All right, now here I've marked the parts which have the English version, okay, of instructions. So mark it earlier in your instructions given to you in your test paper, so that put a tick beside the English version so that you don't miss any of the steps. Okay. So let's discuss the observations and results after 15 minutes of soaking the whisking tubing. Right. So outside the whisking tubing, we have a solution containing iodine. Okay, it's iodine solution, and inside the whisking tubing we have the starch suspension. What is your result after 15 minutes? Now you'll find that the suspension inside the whisking tubing here will turn blue-black in color. Okay, but the solution outside the whisking tubing in the beaker remains the same. So what is the explanation for this based on our biological knowledge? Right, so actually the iodine solution outside the whisking tube here contains the iodide ions and iodine molecules. So these particles, the iodide ions for example, are small enough to diffuse or move through the pores in the selectively permeable membrane, which is the whisking tubing. So the iodide ions can diffuse from the beaker through the pores of the whisking tubing into the whisking tubing itself, right? Then the iodide ions and iodine molecules react with the starch molecules and cause the suspension to become dark blue in color. This is actually a test for starch, okay, a food test for starch. Now, whereas the starch molecules in the whisking tubing are larger in size compared to the iodide ions, and they cannot pass through the tiny pores, which are smaller than them, okay? They cannot pass through the tiny pores of the whisking tubing, all right? So that's why whisking tubing is called a selectively permeable membrane, because it allows the iodide ions to pass through it, but not the larger starch molecules. So the larger starch molecules are prevented from moving through the whisking tubing into the solution in the beaker. Thus, the solution in the beaker here remains brown, yellowish brown, as in the original condition. All right. Now, as a side note, what are the particles found in iodine solution and in the starch suspension, which we are investigating here? For iodine solution, which is used to test for the presence of starch, initially the iodine solution is yellowish brown in color, and then after that, when it comes in contact with starch, the whole mixture becomes blue-black. So what are the molecules in the iodine solution? Now, iodine solution itself contains iodine molecules, I2, which are not charged, and also potassium iodide solution is added to this iodine solution. So potassium iodide contains potassium ions K positive and iodide ions I minus. Okay, K plus and iodide ions I minus in water. Okay, so we have the iodine molecules I2 and the iodide ions I minus. So together, when these molecules combine, or these particles combine, they form the polyiodide ions with the formula In minus, for example, I3 minus and I5 minus in iodine solution. To get the I3 minus, for example, one molecule of I2 combines with one iodide ion, I minus. Okay, that's how you get I3. For I5, it's two molecules of I2 combined with one iodide ion, right? So there's only one negative charge, okay, for each of them. Now, so the particles of iodine that can diffuse into the whisking tubing through the membrane of the whisking tubing are probably the polyiodide ions, okay, which I just state as iodide ions, with the iodine molecules attached to them, all right, inside in the polyiodide ions. So when I state the particles of iodine that diffuse through the whisking tubing, I use the term iodide ions or iodine molecules. Okay, so you can write iodide ions brackets iodine molecules. Okay. Now, on the other hand, for starch suspension, contains the bigger and more complex starch molecules. Okay, so starch molecules are polysaccharides made up of many glucose molecules which act as the monomers. For example, here you see the structure of one starch molecule. Okay, it's spiral in shape, and it can also be a branched, it can also have a branched structure. So here is spiral in shape, and it is a complex molecule, bigger than the iodide ions and bigger than the iodine molecules. So it cannot diffuse through the whisking tubing, as the starch molecules are too big or too large to pass through the pores of the whisking tubing, which acts as a selectively permeable membrane. So for starch suspension, we just mention the particles as the starch molecules. So now let's go on to the paper 3 questions on this experiment from the molecular trial paper. Now, the questions in paper 3 biology usually test some of the science process skills below, as you will see afterwards. For example, they may test you on the observation skill, where you have to make an observation in the experiment. That means to gather information from the experiment using the senses like sight and so forth. So inferring means to make an inference, which is an explanation for observation. An inference is an explanation for the observation. Hypothesizing, meaning make a hypothesis. A hypothesis is the correct relationship between MV and RV. So we have to state the correct relationship between MV and RV. Controlling variables, meaning to state the 3 variables, the MV, manipulated variable, RV, the responding variable, and FV or CV, the constant or fixed variable, and how to handle them. Communicating through the use of tables, graphs, bar charts to present your data, and then interpreting the data. That means we have to analyze the data that we have obtained and be able to give explanations for such data. Okay. Defining operationally, meaning to state the operational definition of a term based on the experiment. Predicting, meaning to make a scientific prediction. A scientific prediction that's based on the original experiment, and we have to know how to explain this prediction. Classifying, meaning to compare and categorize certain items based on their similarities and differences. Experimenting, meaning you have to plan and carry out an experiment successfully, as is stated in the front part of the test paper at the beginning. Then measuring and using numbers, meaning we measure certain factors like temperature or observation or maybe other observations. Okay, measure certain results using tools like using a thermometer to measure the temperature, okay, or the temperature that is increased, and then use standard units to for these measurements. Okay, standard suitable units for certain measurements, such as degrees Celsius, cm, and so forth. So now we're going to go on to the questions, all right? And here, your aim is to try to answer these questions based on the science process skills as accurately, as completely and correctly as possible, okay, in order to obtain the full marks. So take this as an exercise, learning exercise, or just an exercise to test your skills and techniques of answering paper 3 questions, okay? And know what are the components for each of the following. What are the components for an observation, an inference, hypothesis, and so forth. So let's go on to the questions. Instructions. Based on the experiment conducted, answer the following questions. A. State the hypothesis for the experiment. And B. Prepare a table to record your results. So you can try the questions first, write down the answers on a piece of paper, and then check your answers with the ones given here, okay, in order to practice your technique of answering the paper 3 questions. Now, for each question, try to determine the components that are important or needed. All right, like for example, hypothesis. What is the criteria for the hypothesis, or what are the components for hypothesis? Is the MV, RV, usually is the main MV and the main RV from the objective, or sometimes the sub MV and sub RV from the experiment is accepted, okay, as part of the hypothesis. But basically, it's preferable to give the main MV and RV, right, if it is possible. And also, we must have correct relationship. So 3 components, right? For table, we must have the sub MV and sub RV from the experiment, okay, that is used in the experiment, and then the results or the data. All right, so try and write your answers first, and then we'll look at the answers given here. Okay, let's see the hypothesis. Now, we have to determine the MV first, okay, in order to write the hypothesis. So the MV, or manipulated variable, is the size of the molecules, such as the size of the iodide ions and then the size of the starch molecules, which are different, right? So secondly, we have the responding variable, which is the ability of the molecules to diffuse through the whisking tubing. That is the main RV. But to determine the ability of the molecule to diffuse through the whisking tubing, we have to detect the presence of the molecules in the whisking tubing and beaker after 15 minutes of immersing the whisking tubing. Okay, and so another aspect of the RV which is accepted is the color of the solution in the beaker and the color of the suspension, starch suspension in the whisking tubing after 15 minutes, right? So the best answer as given in the textbook. Textbook has a question that is quite similar to this one here, except that no glucose is involved here, whereas in textbook, they put the glucose solution in the whisking tubing, okay, together with the starch solution. Here, there's no glucose solution. So anyway, the responding variable is, the best one is presence of molecules in the whisking tubing and beaker after 15 minutes, okay, and the presence of these molecules can be detected by the color of the solution or starch suspension. Now, next we have the correct relationship, okay? So these 3 parts will make up the hypothesis. So let's look at the hypothesis and see whether it fulfills all the 3 criteria. So Y, which are large molecules, in brackets, starch molecules, cannot diffuse through a selectively permeable membrane, in brackets, right in brackets, whisking tubing, while X, which are small molecules, brackets, iodide ions or iodine molecules, are able to diffuse through a selectively permeable membrane, brackets, whisking tubing. Okay, so here we have the 3 components. The MV are the size of molecules, the large molecules such as Y molecules or starch molecules, and the smaller molecules such as the X molecules or the molecule X or iodide ions or iodine molecules. Okay, then the RV is the ability of the molecule to diffuse through a selectively permeable membrane. Cannot diffuse and can diffuse through the selectively permeable membrane, which is the whisking tubing. Then your correct relationship is that Y is the one that cannot diffuse. Y cannot diffuse through the selectively permeable membrane, but X can. Okay, that means starch molecules, which are larger molecules, cannot diffuse through the selectively permeable membrane of the whisking tubing, but X, which are the iodide ions, and they are the smaller molecules or smaller particles, are able to diffuse through a selectively permeable membrane, okay, which is the whisking tubing. So 3 marks for these 3 components. Next is the table. We have to draw a table to record the result. So your table should have the sub MV, sub RV, and the results, which is the data. So firstly, the location, okay, the solution in the beaker, and then the suspension in the whisking tubing. Okay, so content. For the beaker, it contains 200 ml distilled water plus 5 ml of solution X, okay, or X solution. And then for the whisking tubing, in the whisking tubing, there's 10 ml of suspension Y, should be suspension Y, okay. Suspension is not actually a solution. So 10 ml of suspension Y, okay, but the question here refers to it as solution Y, or Y solution. Now, color of the solution, initial color and final color after 15 minutes. So the initial color of the solution in the beaker outside the whisking tubing is yellowish brown, initially, and then finally it's still yellowish brown. There's no change. Okay, so write down the color. Now the color here is stated as yellowish brown and or brownish yellow. Please don't write yellow or brown. Sometimes you just write yellow or brown. It's not accepted as the observation. Okay, so yellowish brown, this color is mentioned in the SBP paper, answer given for SBP paper, for 1 year, which had an experiment that is similar to this, a bit similar to this. Okay, so I use that description for the color. All right, and then that's the color of iodine solution. Then the whisking tubing. In the whisking tubing, we have the starch suspension. So initially the color is whitish and it's cloudy, right? It's a suspension. Finally, after 15 minutes, it turned blue-black. All right. So this is actually the test for iodine, the test for starch using iodine. Okay. Test is a test for the presence of starch using iodine. So when starch and iodine react together, then we get the blue-black color. Now, if you write blue, it's not right. They usually don't accept the description of the color as blue or black. Okay, it's not accepted. So write blue-black as the color. This is a standard answer given. Okay, for the positive test for starch using iodine solution. Okay, so why did the contents of the whisking tubing turn blue-black? It's because the iodine from the outside of the whisking tubing had diffused into the whisking tubing through the semi-permeable, through the selectively permeable membrane. Okay, we don't use the word semi-permeable anymore here. We follow the textbook is selectively permeable membrane, and according to this question also they use the term selectively permeable membrane. Okay, so the iodine iodide ions or iodine molecules are small enough to diffuse through the pores in the whisking tubing and enter the whisking tubing. Then it reacts with the starch to form the blue-black color. Now let's look at the next question. State the inference based on the observation in B1. Okay, so here's the tip. Now, what is an inference? So you must know that an inference is an explanation, a biological explanation for an observation in your experiment. Okay, to explain the results of your experiment. And the tip is to refer to the objective of your experiment as a guide. So the objective is given at the first part of the explanation, okay, of the question. So refer back to the objective, and here's the objective. You have to carry out an experiment to investigate the movement of substances across a selectively permeable membrane for two different substances which are of different sizes. Okay, their molecules are of different sizes, such as iodine solution and starch suspension, right? So if you look at the observation in B1, in the table of results, you can see that the whisking tubing turned blue-black after 15 minutes, whereas the solution in the beaker did not. So how are you going to explain this observation, right? So it has to do with the size of the molecules or particles, okay, of iodine solution and starch suspension. It's the different sizes of these two, of the particles in these two substances, okay? Okay, so why did the whisking tubing color turn to blue-black, right? It's because the iodine molecules or the iodide ions have diffused into the whisking tubing, okay, and then reacted with the starch to form the blue-black color, right? Okay, so let's look at our answer. Now, we can talk about the Y molecule too, which is actually it's the molecule Y, which is the starch molecules. So the starch molecules or the Y molecules has a larger size, okay? This molecule has a larger size than the pores on the whisking tubing membrane. Therefore, they cannot pass through the whisking tubing, right? Through the pores in the whisking tubing. Whereas the X molecule, molecule X, which is the iodide ions or iodine molecules this molecule has a smaller size than the pores present in the whisking tubing, and they can pass through the whisking tubing. Okay, so that is the answer to explain your table of results just now. So 1 mark for the first part on the molecule Y and 1 mark on the molecule X. Now let's go on to question C. State the variables for this experiment. What is your manipulated variable? So in this experiment, we have the iodide ions or iodine molecules and the starch molecules, which are of different sizes, right? So the manipulated variable is the size of the molecules. And then the responding variable is the ability to diffuse through the whisking tubing. Which one can diffuse through the whisking tubing, right? And from there, it is we determine the presence of molecules in the whisking tubing and beaker after 15 minutes by looking at the color of the solution or the color of the substance in the whisking tubing, right? So your responding variable is the presence of the molecules in the whisking tubing and beaker after 15 minutes. And the presence of these molecules are detected by the iodine test. Okay, so if you ask the fixed variable, then the best answer is surrounding temperature based on the experiment found in the textbook, all right, which is quite similar to this experiment with a slight variation. Where in the experiment, the textbook, 2 substances are placed in the whisking tubing, not just the starch solution, not just the starch suspension, but also a glucose solution. But here there's no glucose solution involved. So next question. Based on the experimental result, explain the operational definition of selectively permeable membrane. So here you have to give the operational definition of the term selectively permeable membrane. And the operational definition means the definition based on the experiment. Okay, so whatever substances were used in the experiment or organisms that are used, you have to mention that also. Okay, so remember the components of the operational definition. What are they? Okay, and try to answer this question first. There's 2 marks here, but try to give a complete answer, right? And then we'll discuss in a while, in a short while. Okay, let's look at the answer. Now, first of all, the components of an operational definition are, number 1, you must know the basic definition. And then in the definition, add in the organisms, solutions, or materials that were used. And then you must have this phrase as shown by the sub-RV in the experiment. And the last phrase is RV is affected by MV. Okay, so our acronym is DORAM, which is D for definition, O for organism, solutions, and materials. R is for shown by the sub-RV in the experiment. And the RV affected by MV is DORAM, is RAM, RAM. RV affected by MV, the A is for affected by. Okay, RV, R for RV, A for affected by, and M is MV. Okay, that's the complete answer. So let's look at our operational definition. What is a good operational definition for selective permeable membrane? Now, a selectively permeable membrane is a material such as whisking tubing. Please mention the whisking tubing, okay? Although it's not mentioned in the model answer, but such as whisking tubing, which we use in the experiment, okay? That allows the movement of molecules X. Now, instead of molecules X, you can put, which allows the movement of smaller molecules X, bracket iodide ions. So you've mentioned the solution used, iodine solution that contains the iodide ions, across them and prevents the movement of molecules Y. So in place of molecules Y, you can put prevents the movement of larger molecules Y, brackets starch molecules, which were used in the experiment, across them. Okay, so a selectively permeable membrane is a material such as the whisking tubing that allows the movement of smaller molecules X, bracket iodide ions, across them. It allows the movement of these iodide ions across them and prevents the movement of larger molecules Y, which are starch molecules, across them. Okay, And this is shown, okay, or as is shown by the dark blue color solution in the whisking tubing. This was our RV, okay, the change in color of the solution, okay, or the suspension in the whisking tubing, okay, after 15 minutes of waiting, right? Okay, so this operational definition covers the DOR of our acronym, but R affected by M, RV affected by MV. We can write that as one final answer, okay? So add on, the presence of molecules in the whisking tubing after 15 minutes, that's the RV, is affected by the MV, the size of the molecules, okay? So altogether will be 3 marks. So 2 marks are given here, so maybe they just want the first part of the answer, okay? But it's better to add on this part, the RV affected by MV, to complete your answer. Now let's go on to the last question. In another experiment, amylase was added to the whisking tubing and put in a water bath with a temperature of 37 degrees Celsius for 20 minutes. Okay, that was the first step. Then only the whisking tubing is removed and soaked in the beaker as in diagram 1.2. So this is like the previous experiment. Okay, so first add amylase to the whisking tubing, put in water bath with a temperature of 37 degrees Celsius for 20 minutes. Then after that only do the experiment like the first experiment, okay, where we soak the whisking tubing in the solution containing iodine. Okay, right. So predict the color produced in the whisking tubing after 15 minutes and explain your answer. So this is a prediction question, and you have to be able to predict using your scientific knowledge, and after that explain your answer. Okay, so prediction question is also based on the experiment at the beginning. You have to refer to the experiment at the beginning. Now let's look at this experiment. So firstly here, the amylase was added. What is the function of the amylase? So amylase is an enzyme, right? Now, so what does it do when it is added to the whisking tubing and put in a water bath for 20 minutes? A reaction occurs, right, between amylase and starch in the whisking tubing. So you must know the reaction, starch plus water with the action of amylase that hydrolyzes the starch. It will form maltose in the end. Okay, so at the end of the 20 minutes, probably all the starch has been hydrolyzed to form maltose. Then there's no longer any starch in the whisking tubing. Okay, so when you immerse the whisking tubing in the beaker containing the solution X, which is iodine, what will happen? Iodine molecules or iodide ions will diffuse into the whisking tubing. Then what happens? Is there any reaction or dark blue color produced? There will be no dark blue color produced because starch is absent, okay, from in the whisking tubing. The starch has all been digested to form maltose. So predict the color. Okay, now notice that the prediction is about the color produced in the whisking tubing after 15 minutes. So the color will be brownish, yellowish brown. Okay, the color of the inside the whisking tubing is yellowish brown. Now please don't just write yellowish brown, full stop. Write a complete sentence, all right? Because incomplete sentences are not so clear in their meaning, so you may not get the marks. Okay, so the color is yellowish brown, 1 mark. Okay, or brownish yellow. Now, if you write brown or yellow, it may not be accepted. So yellowish brown, based on SPP answers. Now, explanation. What is the explanation? Why is it yellowish brown instead of dark blue? Okay, so the enzyme amylase has already hydrolyzed suspension Y earlier, okay, that is the starch, into molecules of smaller size, which is maltose. Okay, and that means the starch is no longer present when we immerse the whisking tubing in the iodine solution. Okay, so even though iodide ions or iodine molecules diffuse into the whisking tubing, there is no reaction with starch as the starch is absent. So the color of the iodine solution will be, or the iodide ions and iodine molecules there will be yellowish brown as in the original condition. Okay, so that's all for this lesson, and I hope that you have gain some useful knowledge from it, and also improve your technique of answering paper 3 questions. Okay, so till we meet again, goodbye and thanks for viewing. Please share, like and subscribe to my channel. Bye for now.